Good afternoon, good morning, good evening, everyone, and welcome. We're thrilled to have you joining us for this webinar on redefining healing, regenerative therapy for chronic wounds. We'll be presenting a wonderful private company today called Aurealis Therapeutics. We're thrilled to have you join us for today's session. What I'd like to jump into is a brief introduction. We'll jump into some introductions from our wonderful and esteemed colleagues at Aurealis, including the CEO, the COO, as well as a leading KOL, Dr. David Armstrong, and the lead investigator, Professor Alberto Piaggesi. So if we could, let's get into some introductions. Aurealis Therapeutics is a biotech developing the world's first multi-target bacterial cell and gene therapy platform. They're trying to redefine how they treat multifactorial diseases such as chronic wounds, cancer, and inflammation. Their lead program, AUP16, has shown superior wound healing efficacy in phase 2 trials and received EMA Prime designation, hopefully validating its potential as a true game changer for millions of patients worldwide. We're excited to explore how this innovative platform could transform cell and gene therapy with scalable, low-cost manufacturing and broad disease applicability, especially in chronic wounds. Today's speakers include CEO Juha, the COO Laurent, the leading KOL from the United States, Professor David Armstrong, and the lead investigator on the phase 2 clinical trial, Professor Alberto. I'll hand it off to the CEO Juha to give a brief introduction, and then we'll go through the rest of the speakers today.
Thank you, Siddharth. Hi, everybody. Pleasure to be here and welcome. So I will start a brief introduction about the technology and the product and the science behind it. So I think that if we start with the needs, so what we are speaking about is I don't see the slides changing. So okay. So if we start with the needs, so we are working with massive unmet medical needs, but also large market opportunities. So if we focus here on the chronic wounds, which in our case typically mean diabetic foot ulcer, venous leg ulcer, pressure ulcers, it's a massive global problem, tens of millions of patients everywhere. Can we go for the next one? So how we think in Aurealis Therapeutics is that we are speaking of complex multifactorial diseases with multiple pathomechanisms involved. It's not only chronic wounds, also cancer and chronic inflammation. And we think that these diseases, they require multi-targeting. That's the key. And what we do know is that current treatments, unless you give many of them different drugs, don't enable that. The other thing is that, of course, that we know that cell and gene therapies, classically, they are very potential. But there is an issue with them because very rarely they can be scaled and the cost of goods is unacceptable. And these are the two main problems that we have in chronic wound care if we are trying to treat them with the cell and gene therapies and having multi-targeting. It is currently not achievable. So this is our solution. What we call it is Aurealis 4-in-1 technology. This is a technology platform. So what you see here in the middle is a lactic acid bacteria, Lactococcus cremoris. It's a biosafety level 1, very safe probiotic bacteria. So we take this bacteria that you have in cheese or yogurt or kimchi and basically genetically modify it to contain 90 copies of plasmid. And what we put into this plasmid that you see the round stuff in the middle is under an inducible promoter, different human therapeutic proteins or drugs, you could say. These can be cytokines, growth factors, chemokines, or almost anything. And this is the platform, so it's a modular one, you can change those. In case of regeneration and wound care, they will be very different, whereas in the cancer, as an example, they will be totally opposite. And what this platform enables is multi-targeting, high scalability, and low cost of goods. And if you think about these products, so they are acting like millions of nanoscale bioreactors in the tissue. First of all, the bacteria activating the tissue, and then the factors that are being secreted from the bacteria are modulating the local tissue microenvironment. In the next slide, I will explain AUP16, what is our lead product for the chronic wound care for diabetic foot ulcer. So this is the AUP16, and how this was designed was having the pathomechanisms, or the, let's say, the healing cascade of the wound healing, the normal healing cascade in mind. What should happen if everything goes right? Of course, we know that in non-healing wound, these things doesn't happen. In healthy patients, healthy wounds, it does happen. So first step, if you think about this, is that the bacteria itself, the probiotic bacteria, once you apply this topically to the wound, it converts the chronic inflammation to acute inflammation. It's a wake-up call for the tissue that something is ongoing and immune cells should infiltrate to the site of action. So that's the first step. And after that, if you think about colony stimulating factor 1 and interleukin 4, that are the two cytokines here that the system is secreting, that is the most powerful cytokine combo to be anti-inflammatory, converting the classical M1 macrophage wrong activation to M2 healing type of regeneration type of inflammation. So this is kind of directing the inflammation to the correct direction. The third step is the angiogenesis and ECM modulation, so basically granulation tissue formation. Fibroblast growth factor 2 and the interleukin 4 are both promoting the fibroblast proliferation, angiogenesis, so helping granulation tissue formation, which is a prerequisite for the epithelialization. And the last step is the epithelialization, so you could say closing of the wound, where also FGF2 is promoting that. So this is how we designed the product. And we have done multiple in vitro studies, animal studies before we went for humans. And then we went for phase 1 trial. And if you go to the next slide, based on the phase 1 trial and the results that we have, EMA, the European Medicines Agency, has granted us the highest designation you can get, actually the only and the first one in this disease, or this indication, DFU, given by EMA to our product in this indication. We also have held a positive pre-IND meeting with FDA. So this all paved the way for the very important phase 2 trial, which was the first time that we were measuring against the standard of care and placebo, and wanted to see the efficacy and safety of the product in a larger population.
So let's briefly introduce Dr. Alberto. Give us a 20-minute introduction of who you are, and then let's get right into the clinical evidence from the phase 2 trial.
Thank you. And I am the principal investigator of this very novel phase 2 trial. I apologize for the voice because I got a cold and that's it, and also for the English, but yes, I will try to do my best. So the study is already ended a few weeks ago, and started more than 1 year ago with the enrollment of 64 patients in 3 different countries, in 10 centers in Italy, in Poland, and in Germany, focusing on near ischemic chronic ulceration with possibly with a long duration, and was designed with 3 arms. One with standard of care and placebo, which was the solution, the diluting solution of the product, of the AAP16 product, and the other 2 arms, one with placebo, with AAP16 administered once a week, and the other one with AAP16 administered twice a week. Patients received standard of care as per the International Working Group of Diabetic Foot Guidelines, and we were they had a very careful designed standard of care with all the other variables homogenized, and they underwent into pre-treatment period of 2 weeks in which they were observed with standard of care, and only those who wouldn't progress towards at least 50% reduction of the wound would be enrolled in the study. Then they will be treated for 12 weeks and follow up until 20 weeks, where the primary endpoints were set. The patient were then followed up to 12 months with a long-term follow-up to see what happened afterwards. But we are now at the phase of having the results up to 20 weeks. The endpoints were safety, of course, and efficacy. There were no difference in safety issues, and we see what per protocol were the major endpoint, the principal endpoints, and you can see a reduction of twice reduction of increase of healing rates in the group treated twice a week with the AMP16, but also despite this did not reach significant difference, but it was twice compared to the standard of care. And if you select and if you reduce the number of patients and you focus on those who were only those who were chronic patients, if you give me the next slide. Focusing on those whose duration were more than 3 months before enrollment, again per protocol. This means all the patients that completed the treatment. You can see that the healing rate raised to threefold compared to the standard of care, reaching significance with the chi-square test a probability of less than 0.043. More interesting from our point of view was another aspect of the study. If you move to the next slide. This was a secondary endpoint. It was the wound area reduction in the cohort of patients in which the wounds were chronic, the wounds that were already there since 3 months before the enrollment of the patient. And you can see how the progression of wound reduction, or wound area reduction, clearly differs in the patients treated with APU16 twice a week compared to placebo. And you can see that at 20 weeks and the set where the endpoints were set, the reduction was extremely higher, was 6 times the rate of was the figure of the placebo of the comparator. From my point of view, is also interesting the fact that the two curves start to diverge early in the process. Already after 4 weeks of treatment, you can see that the difference between the two groups diverge, and this divergence is much more intense in the treatment group compared to the placebo group. The slope of this curve is much more steep than the other one, who gets a less steep shape. And again, at 20 weeks, we at the limit of significance with 0.054. Can move. And we have some cases just to show you which kind of patients we have in the group. There were, as I told you, neuroischemic patient very chronic. You see these patients in the placebo group already underwent the amputation of the first and the second toe because of an infection in the toe, but then developed a consequence, a transfer ulceration on the first metatarsal head, but didn't heal despite the good standard of care. And this other patient is another very difficult to heal. Can you put the previous slide? Yes. This patient has an ulcer in the heel, which has demonstrated to have a very difficult to heal location and a very slow trajectory of healing. In the 1 per week group, you see that in 20 weeks was healed, and it was, from our point of view, from a clinical point of view, a very encouraging. In the twice a week group, you see again an ulceration, a chronic ulceration on the second ray, on the second metatarsal head, and that progressed very fast to heal. At 12 weeks was almost healed already. And the last, but not the least, from my point of view, is this very chronic lesion, dorsal lesion after debridement and intervention in these Charcot patients, in which the lesion was there since 1 year and didn't move in this period, while in 20 weeks with the administration twice a week of the placebo, of the treatment was healed.
Thank you, Dr. Alberto. Briefly, could you just tell us why you believe this could be game-changing compared to the standard of care?
You see, we already apply standard of care extensively in this patient that we enrolled for a long time before they got into the study. And the difference we observed from a clinical point of view, despite being blinded according to the design of the protocol, we knew that the ones in the treatment group got a different curve from the others, and the impression from a clinical point of view was striking since the very first phases. Beyond this impression, beyond this clinical impression, there is the scientific hypothesis that is baking this approach, which targets different biological mechanism of the biology of the wounds, and from my point of view, as a sort of an integrated biochemical approach to a process which up to this moment has been frozen in a chronic inflammatory state. And we can see also the quality of the tissue that we observe from the change of dressing and the progressing of the cases. We observe a visible changing in the quality and the quantity of tissue which was newly formed by the inception of this strategy in the treatment.
Fantastic. Thank you, Dr. Alberto. I'd love to hand it off to Dr. David Armstrong to talk a little bit about the commercial perspective and the real-world evidence and impact of this therapy. If you could, Dr. Armstrong, just give us a brief introduction about yourself.
Well, ladies and gents, it's fun to follow my longtime friend Alberto. It's great to be with you guys. Thank you for letting me play hooky from what it already is a crazy day. I've already been in the operating room all the night taking care of this problem. So you want to talk about an unmet need? You guys can come all help me with this unmet need here in Los Angeles. I'm just a foot doctor here. I'm purportedly a distinguished professor of surgery and neurological surgery here at Keck. But I hopefully will convince you that this is an unmet need within an unmet need. I mean, very few people care about diabetes, even on this call. Even fewer people care about feet. But this is extraordinary. It's one of these unmet needs within unmet needs, because people have been blowing this off for the longest time. And it's to our peril, because now if you look at these next data from a study we had you can go on the next slide, from a study we had in the New England Journal not too long ago, we looked at all the best available data every second now around the world, someone gets a wound. The clock is ticking back, not forward here. It's bad. Half of these get infected. 20% of those infections end up in hospital, which is now why about every 20 seconds someone gets part of their leg off or their leg off. And this is a tragedy, and this is a problem we believe that can be completely eliminated, not only with teams, but with technology. And you're seeing some exciting stuff from Professor Piaggesi, because we think we can marry a pretty old technology like a bacterium with a new sort of platform technology and engineering that thing, because we think time's up for this whole thing. We can really make a difference. So let's go on to that next slide. Speaking of an unmet need, these data from our team and from others, including Alberto's, suggest the most conservative numbers suggest that there's about 15, 16 million wounds every year. You might even be able to increase this by 30%. Care right now is all over the place. It's not great. And healthcare systems, public payers now are struggling with this. It is occupying sometimes in the single digits of the entire healthcare budget, just for feet, not even for all the other wounds. It's extraordinary. Next slide. And it's not fair to compare one terrible thing to another, but again, I think we all care about cancer, and we all have family members who have survived or succumbed, but very few of us care about feet or diabetes until maybe now, or when we're talking about this unmet need. But what we know now, remarkably, and I'll share all this with you later, the treatment of cancer right now is more expensive in the United States, treatment of diabetic feet, than the 5 most expensive cancers. And there are plenty of people right now on calls talking about breast cancer and colon cancer, but we happy few are the only ones talking about feet. Again, unmet need within an unmet need. Mortality now with diabetic foot complications is worse than all but the worst cancers now. 30% of people with diabetic foot ulcers will be dead if they get an amputation. Up to 80% will be dead. That is worse than aggressive lung cancer and even some pancreatic and brain cancers. So this is, again, just as a comparator, breast cancer is around 15, 18%. Next, this is just a review we did. You can keep going. These people get wounds, folks with diabetes, because they lose feeling. So it's a silent problem. They literally wear a hole in their foot like we'd wear a hole in a sock. That's what happened to the patient that I was in the operating room overnight. That's what happens every second. Next slide. And they literally just get a blister. That blister gets inflamed. You hear about inflammation and the anti-inflammatory capacity of this platform. Here you are. Next slide. And sort of, you can go on to the next slide. You can go on to the next one. But let me show you this. I know it's a busy slide, but it's important. These are the best available data from our team and others. After people heal, are they really healed? Because at about 1 year, 40% of folks are going to get a recurrence of a diabetic foot ulcer. It's about 2/3 at 3 years. It's about 3/4 at 5. So recurrence in this population is not only common, you can go to the next slide, it's likely. So if the foot in diabetes, you can go to the next slide, is a little like cancer, then maybe when folks are healed, maybe they're in remission. And this seems like just a play on words, but it is not. It fundamentally changes the way we treat all of these chronic diseases. And it fits so perfectly with this therapeutic, which has multi-platforms in its anti-inflammatory capacity. So which is what's particularly engaging on this area. And we always say now that nothing ruins a good surgical result like follow-up. I literally said this to my fellow just a few hours ago, because you feel like you've helped someone in the operating room, but that's just the start. We're treating these folks. It's not a one-time thing here. It's not like an antibiotic or a pain pill. This is from the alpha to the omega. So this should be looked at more like a statin, or more like a cardiovascular therapeutic, or even a cancer therapeutic than just an antibiotic, for instance, let's just say. And we now have data suggesting that recurrence in this patient population is now at least as bad or worse than aggressive cancers. That's the big idea here. We really believe that we can make a difference together when we marry team, like the great ones you have with Professor Piaggesi, with a little technology, which is what you have already heard about and you're probably going to hear more about. Thanks a million. I really appreciate it. Thank you for letting me play hooky again.
And before we get into Laurent's part on the strategic vision, Dr. Armstrong, could you emphasize a little bit about the quality of life gains and impact on healthcare system efficiency? I'm assuming there's billions of dollars of associated costs with it, and the impact on quality of life.
That's a great question. Even if that wasn't a great question, I'd say that's a great question. But FYI, that is a great question. So quality of life right now for folks with wounds is worse than if you look at all of these other chronic diseases that we know are important. We look at World Health Organization data. This is like you've had a recurrent breast cancer or something or other. Most of these patients are depressed. 40 to 60% are depressed with the most conservative data. This takes people out of being what they're doing, whatever it is they're doing, and puts them forever sort of at the mercy of other people and a system. If we can get these folks healed, there are data from Professor Peter Jesse and from others, including us, that show that you can dramatically reverse that. And that is just life-affirming stuff, because you can help these folks move through the world a little bit better each and every day. And that's something that's easy to look at on a slide and hard to do in real life. But just because it's hard doesn't mean it's not worth a life's work. And this therapeutic can potentially be really helpful there.
Solid answer. Thank you, Dr. Armstrong. Laurent, I'd love for you to introduce yourself briefly and get into the strategic vision and pipeline expansion.
Don't mute yourself, Laurent.
Yeah, am I still muted, or can you hear me?
Oh, we can hear you.
Okay, perfect. So I'm Laurent Decoré, Chief Operating Officer. If you go to the next slide, not only we are really proud of the results of our AP16 chronic wound program, but as mentioned by Juha earlier, our platform is extremely modular, and beyond AP16 chronic wounds, our second modalities with cancer oncology, with our product AP55, with already great in vivo results. Next. AP55 is based on the same bacteria. The genetic modification and resulting mode of action is almost the opposite from AP16. So here we fight, we don't heal. So the endogenous factors, TLR2, TLR9 node2 agonist, induce cancer cell death and reduce tumor blood supply. The added factors, human interferon alpha and human IL-15, activate innate and adaptive immune system, DC cell, macrophage, etc., killing more cancer cells. So AP55 is really a powerful multi-target anti-cancer agent. Next. The mode of action of AP55 is already validated by multiple approved products on the market, such as Live BCG, MePAC, Piquipanil, Astiladrin, or Anktiva. The different components of AP55 mode of action have been demonstrated in vivo. You can see different graphs, but even more importantly, we have demonstrated massive tumor reduction and survival benefit in ovarian cancer in vivo studies. And in less than 12 months, we will enter the clinic with the first in-human studies. So next slide. If you consider AP16 and AP55, you compare the Aurelis platform to other cell and gene therapies, such as viral vectors, CAR-T, for example. Not only we are the only one allowing multi-targeting, addressing all these complex modes of action, but also we are the only one with high manufacturing scalability and low cost of goods. We talk about $1 per vial in manufacturing cost. Nothing you can hear about that from CAR-T or viral vectors, for example. This is extremely important when going to the market in very large markets, such as chronic wounds, for example. Next. We have raised 60 million so far, and now we're opening 45 million Series B. 50% of the round is already committed by the lead investor. And this round will enable three things. First, AP16, a global pivotal trial in diabetic foot ulcers, as well as an additional phase 2 study in venous ulcers. Secondly, AP55 multiple investigator-initiated trials and starting the phase 1 study. And finally, expansion of our manufacturing capabilities for phase 3 and commercial process. So that's really a very quick overview of our Series B and investment case, but I think we can now move to Q&A and discussion.
Thank you, Laurent. I want to first thank all of our speakers for joining us today from all over the world. We have Juha, Laurent, Professor Piagetzi, and Dr. Armstrong, and to everyone who's joined us today. We hope this session gave you a clear view of what they are trying to accomplish in redefining chronic wound care and advancing a truly scalable multi-target cell and gene therapy platform. What I'd love to do is we have a few questions that have come in. I'd love to get into those. Before I do that, anything you want to say, Juha or Laurent?
No, I think it's, first of all, thank you all for excellent presentation, Alberto, David, and kind of showing us the real problem. I mean, sometimes when we are like 24/7 developing the drug, we tend not to see the patients always. So it's also for us, not just the general audience, to refresh the kind of the memory about the real problem out there, why we are doing this. And also emphasize that even though we have been committing to this, developing this drug since the inception already for 10 years when we designed this product. I mean, Alberto, David, you guys have been working for decades to make the lives of these patients better. So, but I think that what is most important here is that now we have completed phase 2 trial. We are getting ready for the pivotal trial, and we are getting closer and closer to the market of this product. So I think it's going to be really exciting next few years about doing the pivotal trial and seeing the results of that and getting this treatment for the patient. So certainly this is extremely motivating for drug development. It's a long game. It's a long game. But when you are seeing the target, you are seeing the goal, I think we start to be there.
If I may comment, I think that's one of the aspects that's it's important to remember is that the number of patients is increasing, and the access to technology is what is more and more an issue in our clinics, because despite the fact that other solution may be effective, the number and the recurrency rates of this patient are such that the easier and the more accessible is the technology, the higher is the benefit that the patient can get from the technology, irrespectively from the complex, the easier, and in this easiness also the complexity of application of the technology, because the way in which we treat this patient is not different from a normal dressing change. And this may play a difference in when you choose the best possible, I would say, efficacy complexity ratio of a treatment. And this is something that we appreciate very much in this experience.
Thank you. So a question that's come in, I think this is a good segue. This is for Juha. As you know, the wound care market is so heterogeneous and so broad. The gene and cell therapy market is so heterogeneous and so broad. I mean, there's so many platforms in development today for cell and gene therapy. What makes your specific bacterial cell approach fundamentally different, both from a scientific perspective as well as from a scalability standpoint?
Well, I think it's quite obvious. So what we have seen, because we come from the background of working with these cell and gene therapies for decades ourselves. So what we see is three things. Is first of all, very rarely you can enable multi-targeting. So Aurealis is based on this technology, enabling 4-in-1 targeting. So targeting up to 4 different targets in the same API, same active pharmaceutical ingredient one. That's number one. I think efficacy is driving everything. Of course, feasibility, like Alberto mentioned, is very, very important. But then the other two things is always the cost of goods and scalability. So it doesn't really help if you cannot serve tens of millions of patients. If, like in some of the cell and gene therapies, viral vectors, CAR Ts, others, you have really issues of even providing the market enough of the drug. We have no issues like that. We can already produce millions of vials in one production run. And then cost of goods is of course important, because if you think about the rare disease and cancer, yeah, you can price it for $500,000 per patient, maybe, probably not in the future, but especially in indications like wounds and all over the globe, let's say even Asia, China, and other countries, you can price it for $50,000, $100,000 per patient. You are going to be at maximum around $1,000, $2,000, maybe $5,000 per patient. So basically, with our product, with this cost of goods, you can still have a extremely profitable business by serving everybody on every single market. So efficacy, cost of goods, scalability.
That seems to be a pretty good value proposition, right?
Exactly.
So another question, I think both for Juha and Laurent, you've kind of reached the point where you have both EMA prime designation, you've completed a phase 2 study with positive results. How do these milestones, de-risk the path forward, right? The path to approval, the path to getting this therapy to patients, and what are kind of some of those key either clinical or regulatory inflection points that the folks should watch out for the next 12 months?
Well, if I may start, so first of all, we have met EMA, FDA, and CD in China. All of them endorsed this, and actually encouraged us to file for the fast track designation. We already got one in Europe, and we will hopefully soon get from FDA when we file the IND for the pivotal trial. So I think it's all about getting the clinical data, the clinical evidence. When we developed this early on, everybody was saying that, hey, yeah, bold approach, but wound care is challenging both medically and commercially. But everybody knows that at some points, typically at phase 2 data, the risk reward ratio is start to be favoring you. So when you have more and more clinical data, so I think that we are there now, and this is the right time to join this company as an investor as well. So I think that's certainly encouraged by the pre-IND meetings and scientific meetings with the different authorities around the globe, especially FDA, EMA, and CDE.
Wonderful.
If I can add one element to that, we have already developed and proven health economic benefit and cost effectiveness of the product based on phase 1 data, now based on phase 2. We have interviewed payers in US, in Germany, in UK, who have confirmed the reasonable pricing assumptions we have for the market and ratio of the benefit. And another milestone, we're pursuing an early access program, and we have good hopes that this product could be available as early as 2027 in countries like France through early access programs, also contributing to real-world evidence gathering.
And talking just about kind of the clinical path forward, when do we look to initiate a phase 2b/3 clinical trial? And will it be, the EU only? Will it be worldwide? Will it be US? What is the team thinking there?
It's going to start next year. I know, of course, depending on how you define start, but having the pre-IND meeting submission next year. We also plan to start before end of next year the pivotal trial, and this would be USA, Europe, and China. Phase 2b/3 trial all over.
Wonderful. So moving on to maybe some clinical questions. This is both for Dr. Alberto and Dr. Armstrong. I'll give the first one to Dr. Alberto. In the course of the development of foot ulcers, chronic wounds, when do you see the best timing for applying AUP16? Given all the other surgical interventions and treatment options that exist in this world.
Yeah. Based on the experience we did in the study, in the benefit that we observed, as we said, is in the chronic phase of the foot, in which you may find different clinical patterns of diabetic foot. You may find a post-surgical foot, which already has been debrided, but still have some difficulties in progress. You may find a contaminated ulceration, which is not infected, but in which the presence of bacteria is enough to determine a slowing, the slowdown of progressing of the biology of the wound. You may find, as we said, as we observed in our study, the mildly ischemic foot, in which ischemia is not so critical to be an indication for revascularization, but is still enough for interfering with the normal processing of healing. So there is a variety, a catalog of possible indication that should be tested and explored but in which potentially the inception of this kind of technology may play a difference.
And do you see AUP16 changing the standard of care for chronic wounds in your real-world practice?
For sure. If it would be available for a wider number of patients, because we were committed to do the study, so it was a very selected number of patients because of indication for the study. But if we would be in the condition of applying to a wider plateau, a wider cohort of patients, we would sure use it extensively.
And thank you, and I appreciate your clinical insight. The next question we got, and I think this one's going to be for you, Dr. Armstrong. So given the very large annual cost of chronic wounds worldwide, right? Whatever article you may be reading, some estimate as high as $100 billion. How do you think, or how should we think about the addressable market and kind of the health economic value of this therapy compared to kind of the existing wound care products we have, all the way from kind of the more tech-focused products like wound wax to the topical gels and the dressings, and I mean we can go on and on.
Yeah, I think we've been collectively nursing these wounds really well for a long time. In other words, there have been kind of commoditized dressings that absorb things, that maybe reduce pain a little bit, that are in cabinets in hospitals, from South Asia to the North Africa to North America to Europe. Those are mind-numbingly dull, but somewhat helpful. But those create a lot of noise. It's only up until fairly recently that we have begun kind of, if you will, doctoring these wounds. And it's only been the last generation that this has started to happen. So this stuff happens slowly. But you mentioned negative pressure wound therapy. There's cell-based therapies and others. This has the potential, just as you heard from Professor Piaggesi, to be really transformative because it is literally and figuratively a platform just by itself, with nothing inside of it, even if it wasn't working. It's probably therapeutic because it's lactobacillus. It's a kinder, gentler bacterium that could be fundamentally modifying the milieu of the wound. But then adding things in can create literally multiple platforms, not just from this, but in the future, that could be marrying with things that we're working on right now that are companion diagnostics and theranostics. I was in the operating room and I was using special loupes that allow me to see bacteria, transilluminate bacteria, and that allowed us to do image-guided debridement. Things like this, you heard Professor Piaggesi touching on, can tell us when to start and stop things. Many of these things, we don't want to be wasting money putting something like this on something that needs to be sort of surgically reset, just like a computer needs to be Control-Alt-Deleted. And as that wound is rebooting, there is a flowering now of very inexpensive companion diagnostics and theranostics that could help us with therapies like this and others. And I see in the very near future, not just like, I'm talking like over the next 3 to 5 years, multiple therapeutics being used at the same time, just as we are very comfortable using using a statin by measuring cholesterol, along with a diuretic by measuring blood pressure. This is happening now, and this is coming in at the right time. And you see the enthusiasm by the regulatory agencies already, just with phase 2 data.
Yeah, thank you. I think you gave me a little bit of PTSD from my surgical training, but it seems like this therapy is addressing some of the root causes and not just the symptoms that we see in chronic wounds. So
100%.
Your insight. And I think this is the last question we have. This one's probably for you, Laurent. As you advance into kind of late stage trials, right? Potential global partnership discussions, how are you thinking about the capital strategy? Do you want to remain private? versus an IPO or M&A, and what does success look like for the company by, let's say, 2027 or 2028?
I mean, I can start, and you probably will build on that. But basically, we are a private-owned company, and we've been successful like that, and we are operating as a B2B. But on the other hand, we know a number of investors really want to see a clear path towards IPO, and we probably would see 27 probably as the right timing for us to go a US IPO to further expand, and in parallel also ensure that a global partnership with, for example, a global wound care company would enable to amplify the commercialization of AUP60. You have anything to add?
I'll just fully echo on that. This round that is right now ongoing, Series B, I think this would be our last private round. So I think that's a point, and then we would aim for an IPO in late 26, early 27.
Perfect. Well, thank you again to all of our speakers. We look forward to sharing upcoming clinical milestones and continuing the conversation with interested partners as well as investors. Thanks to everybody, including our speakers from all over the world, for the time and the engagement. Everybody, you guys have a great rest of your day.
Thank you very much.
Thank you.
Take care.